Today, we're going to talk about the semantic hierarchical exploration of large image datasets. It's really hard to explore large sets of images. To simplify this, we typically put our images into libraries, such as Apple Photos, which you can see here. These libraries organize the images for us, for example, by date, as you can see here. However, maybe I don't always know when I took a picture that I want to inspect. Furthermore, this includes a lot of scrolling, so I have to scroll through all the dates before I get at the image that I want to look at. To make images explorable in different ways, there exist approaches like this map-based approach, where I can zoom into regions that I know I took some images at and get all the images that I took around this location. However, this is only one categorization, namely location-based categorization. And with modern smartphones and modern devices, we take a ton of pictures. So we really want to explore them in multiple different ways. The same is true for modern image datasets, such as machine learning datasets, that typically include very large numbers of images. So we argue that people should be able to explore images using their semantic context, instead of just metadata that's readily available. And to show you how we did that, Christian's going to give you a demo of the system that we've built.
This is Hierarchical Image Explorer. Here we can see the ImageNet dataset classified by a machine learning model. Each honeycomb in this view represents a group of images within this region in the embedding space. A sparse region where we are not grouping together images can be seen here. This single image is not drawn as a honeycomb, but a square instead. To get a better understanding of the images and the dataset, aggregated into one honeycomb, we can click on a honeycomb. Here we can see the distribution of all images and the labels of those images within this cell. We can even inspect regions within the visualization by using the lasso tool. We can also compare two regions of the plot. If we identified regions of interest, for example the regions with the dogs here at the right, we can zoom into this region. And we can see while zooming in, we subdivide the honeycomb cells, allowing for a finer inspection. We can zoom in even further until we see individual images again. The minimap here at the bottom right helps us to maintain an idea of where we are within the embedding space. Let's now say we navigated this dataset for a while, and we are now interested in particular patterns within that dataset to evaluate our model. Let's say we are interested in images that are misclassified by the model. To investigate this, we can use our filters. We can add a filter, and we filter for images where the label is not the same as the prediction. We can then apply this filter, and we now see only mislabeled images. We can even chain filters together. Let's say we are interested in other examples of images with this particular label. Then we can add another filter and filter for images with this label.
You just saw the front end of our approach. So what happens behind the scenes? Well, the user first supplies images and their associated metadata. We analyze the image semantics and project them into a 2D semantic embedding space. Here, images with similar semantics are placed close to each other, while less semantically similar images are placed far apart. Of course, many images that are close by causes overlapping images. To deal with this, we utilize the hierarchical level of detail approach that groups similar images in the embedding space, which Christian just demoed. There are many types of semantics that can be relevant for different use cases. We supply 3 different options. We have the embedding based, where pre-trained machine learning model creates a semantic embedding of the image. Here, vision models have been shown to learn high-level concepts that can map to semantics that we humans relate to. We have pixel-based, where raw pixel values are used for each image. This is a low-level approach that only separates images by their color and is mostly used as a fallback. Finally, we have vector-based, where users can optionally provide a custom vector for projection together with the dataset. This method makes our pipeline highly customizable. Independently of the embedding method used, we always project the high-dimensional input into 2D space to allow for visual exploration. Computing the 2D projection can be slow and require extensive amount of memory, so for large datasets, we compute it based on a subset of data. We say this computed projection model, which means that new images can be added quickly. Once the embedding computations are done, the embedded image positions are discretized to a regular honeycomb grid. We chose a honeycomb grid rather than a square design to allow for dense packing while optimizing the distance of an image to its grid cell center. One can think of this as a trade-off between squares, which perfectly pack, and circles, which minimize the distance between points and their cell center, but don't pack optimally. Higher hierarchy levels will represent a large embedding space, so we use the image closest to the cell center as a visual representation of the images in the cell. The border color of each cell can be chosen to represent one of the metadata fields in the dataset. Groups of images, in other words a grid cell, can be compared by selecting one or more items. Their corresponding metadata are automatically aggregated and their distributions visualized. Advanced filter expressions can be formulated to allow exploration of particular aspects of the data. Here we inspect 2 different classification models, Inception v3 and VGG16. Multiple filter expressions enable the display of misclassified images by both the Inception v3 and the VGG16 models. In this particular part of the embedding space, we can see that the misclassified images are either sports cars or close-ups of the front of the car. This information can be reported back to the quality assurance team for improving the dataset or model. The combination of advanced filtering, visualization, and different embeddings means that the method can be used in a wide range of application scenarios. Here we see an example of blood smear slide images of malaria cases that have been semantically embedded into a space that separate parasitized and uninfected cells. The underlying model for the embedding space have been trained to classify malaria. In the paper, we describe how such an embedding could be used for medical training to, for example, demonstrate which malaria cases that require special attention. These special attention malaria cases are illustrated in an inset image, which shows a zoomed-in view to a finer hierarchy level that allows for inspecting these individual malaria cases.
So that's HIE. Our approach is scalable, semantics-based image exploration. HIE is based on multiple concepts, such as different hierarchy levels, a sophisticated embedding pipeline, representatives for image groups, and powerful filters to enhance your exploration capabilities. The best of it, HIE is really easy to use with your own data. Just check it out at the GitHub link that's shown here.